Hey, what's up everybody, and welcome back to The Sanctuary. I'm your host, Professor C, and we're going to keep on trucking through some A&P today. Specifically, we're going to look at the sliding filament theory and some of the details involved with actin and myosin. Let's check it out. Okay, we can start off the talk with a rather morbid discussion, but why do dead bodies become stiff? And I know some of you may be screaming rigor mortis is the answer to that, but what specifically is occurring in the body that allows a dead body to become stiff in a rigor mortis event? Well, to answer that, we're going to have to go through our third part of our excitation contraction coupling sequence here and get down to the nitty-gritty. Finally, we get to where the muscle is actually going to shorten. On the previous two talks, we've generated an action potential along a motor neuron, this big yellow thing up here that I'm drawing the waves on, and that caused some calcium to come into this terminal knob and put acetylcholine in a vesicle and send it out into this neuromuscular junction where it bound with a muscle fiber waiting on the other side. And where we ended the second part of that story is that action potentials were then generated here along the sarcolemma after the activity at that motor end plate. So we ended with a problem, and we said that's great that we got some action potentials over here, but we don't want them there. We need to get that information down deep into the muscle, and it says that right here. We need to send the information deep and stimulate the sarcomeres, and you can see a lot of them in here. Here's a Z line, if you remember, and here's that junction between the I band and the A band, and you can go back and check out talk number 85 if you want to see the details of that. But we need to make these Z lines come together now. We need to make the sarcomere shorten, and the action potentials are not going to be the thing that stimulates it. So let's start the story with the muscle triads that we saw in another talk as well. I believe that was also in 85. So T-tubes, also known as transverse tubules. These are the extensions of the sarcolemma here, the cell membrane of this muscle fiber, and that's what this big giant structure here is, and the whole picture is one giant muscle fiber. And we know from our previous talk that the T-tube reroutes those action potentials deep. Now again, those action potentials aren't going to stimulate the sarcomere directly, but they will stimulate these cisternae. I'll just put two C's here, and it says right there, they will stimulate the two cisternae that flank the T-tubes and the broader sarcoplasmic reticulum. And you can just see right in here and right in here some of the extensions. That would of course make a network all the way throughout here. And that structure, both the cisternae and the SR, loaded with calcium. And so when the action potential comes down the T-tubes, it causes the other structures to spray out calcium. And that's what this red ball structures are supposed to be here. It's calcium, and it's being pushed deep where we can get to those microfilaments, actin and myosin. So let's see the details here. It's going to be called conceptually the sliding filament theory, and it's called that in almost all of the textbooks. And what we have to start off with first is what happens if nothing happens. And we saw this calcium come out from the cisternae and the sarcoplasmic reticula at the end of the last slide. So what we need to do is set this up and see what it looks like. So I'm going to put a long thread here, and I'm going to call it myosin, just so we know for the future that's a strand of myosin protein. And I'm going to put a head on it because we know it's got kind of a golf club head that sticks out like this. And let's just show one of them to make it simple here. Now, where's actin? Well, actin's over here, and I'm going to draw it very simply as well. I'm not going to draw it as the twisted helix, but I will show some binding sites along here, and that's where those myosin heads want to go. So he wants to get in there and attach to the actin. So I'll put actin here so we know what we're talking about. Now, it can't though. And the reason it can't is because a ribbon is going to be going over it like that and blocking it. And that ribbon is called TROPOMYOSIN, and it wraps around the actin. So it's saying here at low calcium concentrations, the myosin heads cannot attach because tropomyosin has blocked the binding sites on the actin. So if you're not going to attach, you're not going to be able to pull it. and you can't pull, you can't get shorter, so we absolutely cannot contract if myosin and actin cannot hook up. So we're going to talk about how they hook up in a sequence of 4 events. And this is a 4-event cycle, and once you get to the end of the 4, it'll start at number 1 again, and over and over and over and over. So let's take it one by one. We can start with, well, attachment. Okay, let's look at actin again and remind ourselves of some things. We know there is some binding sites here for the myosin heads, and we know there's going to be a ribbon that goes around there called TROPOMYOSIN that blocks it, but we have to think about another thing. There's a regulatory structure here, and it's called troponin. And we kind of told a story way back in probably talk number 85 also, that at some point calcium can bind to troponin and turn it on, so to speak. So that's what we're going to see happen first. Now again, where'd the calcium come from? It came from the flooding of the cisternae and the SR when the T-tube sent some action potentials deep inside the muscle. So there's the calcium, and it's going to bind to the troponin, not the tropomyosin. Tropomyosin is the ribbon. But what happens is when the troponin binds, it kind of twists actin in space, and it pulls that ribbon out of the way. So instead of being in the way now, the ribbon is open. Like that. The ribbon's been shifted over, if I can show it like that. And the binding sites on the actin for the myosin heads is open now. So I'm going to draw the same little critter I drew before, like this, and there's my myosin. And in this first event, he's going to have 2 little things attached to him. He's going to have an ADP molecule, adenosine diphosphate, and then hanging off here will be what's usually written as a PI, an inorganic phosphate group. And I've talked about this in some very old chemistry lectures if you want to scroll back and do some of those old ones. But this is what it would look like in the attachment phase. Calcium has come in, it's bound to troponin, it's twisted actin, and it's pulled tropomyosin out of the way. The head is attached, but it's not done anything yet. It's just simply attached. So this ADP and inorganic phosphate that are stuck to that puts those myosin heads in a high energy configuration. They're a loaded pistol, right? The pistol is already there and it's already cocked like an old cowboy pistol, right? It's ready to fire off, but it's not fired yet. But it has now attached to the actin. So if we can get the gun to fire now, being that it's already cocked from that high energy configuration, we should be able to get, say, a Z line maybe that's over here to slide over this way. So the second event here in the 4 is called the power stroke. And what we're going to do is, well, we're going to uncock the weapon, right? It's going to fire off now. And what we're going to see is something like this. We're going to see our actin up here. I'll just put A for actin at this point. The important part is there's the binding sites for the myosin heads. Let's put one of those heads in here that we just saw, but now this thing is going to be cocking over just like the cock of a gun is fired off. Now in order to do that, we're going to have to dump off ADP and that little inorganic phosphate group that was hanging out with it. And so part 1 here of the power stroke is get rid of ADP and the inorganic phosphate, and this will cause the head to click forward. So the myosin heads will pivot or cock or ratchet forward, and they will pull the actin with it. The actin now, the whole molecule, will be pulled this way. And again, somewhere down the line over here, there's going to be a Z line that's the end of the sarcomere, and it will cause that Z line to go this way. So the sarcomere will shorten at this point, and we've done a muscle contraction, right? It might be a tiny, tiny, almost unobservable one, but here we've got at least one part of a fiber to shorten. Okay, now that we've lost our ADP and our inorganic phosphate, that head wants to recharge. So here's our actin, and I'll just kind of sketch it very quickly like this, actin. And as long as there's calcium present, the tropomyosin will stay off of those sites. But at this point, the myosin will detach. Okay, so this myosin head detaches at this point, and that ADP is replaced by, sorry, I wrote the wrong letter there. The ADP is replaced by an ATP. Put a big flash around it so we know it's ATP. So the myosin heads are released, and a new molecule of ATP is added to replace what has been lost. Now we haven't recocked yet, but we're about to. We've recharged, and we're about to recock the gun for our 4th step, and then we can attach and do it again. So the recocking phase involves breaking ATP now that was just recently added into ADP and organic phosphate again, just like it looked in step 1. So this process is called hydrolysis, and I can show it this way. Here is my actin molecule again. Here are the binding sites. Again, as long as calcium remains here, then those binding sites will remain open. Here's our myosin, and I put our head here. Again, we're not attached again, but I have recocked myself. I have pulled myself back into position. And I know, I don't think recock is an official term, but you hear it often, kind of like talking about a gun. Inorganic phosphate is added. That ATP has been broken down into that, and ADP, right? And this, of course, is called hydrolysis, this process of breaking it apart. This 4-stage cycle will then continue. It'll go back to step 1 where we attach, as long as it says, until calcium is pumped back into the SR, and this usually occurs when the stimulation of the neurons ceases. Okay, there's a term called a latent period, and it is between the time, or is a time between, I should say, the initial action potential on the motor neuron, all right? Not the initial action potential on the muscle fiber. So way back at the beginning of our story, when we started in episode 86. So all of 86, 87, and 88, all that stuff that we talked about, all the way through the end of this talk 89, that's called a latent period. So the initial AP to the contraction of the fiber. Tell that whole story in about 5 milliseconds, and that's how fast the body can do it, even though, of course, it would take me about an hour to tell that entire story with all the details. Okay, so back to our rigor mortis. Why does a body become stiff upon death? Well, think about it this way. You have to go back to that 4-step series of actin and myosin doing their sliding filaments. When cells die, they don't continue to grow. There's a mythology there, but it's not true. They start to deteriorate very quickly. So when cells deteriorate, like a muscle cell, calcium will leak out of the sarcoplasmic reticulum and basically flood the internal surface of that cell. Myosin heads will attach, right? They've already been cocked. They're already ready to go, but they will still attach and pivot. But they cannot be uncocked, right? Because there's no ATP to do that recharge step that we talked about. They can't recharge the ADP and the inorganic phosphate that they use to cock themselves over. So they basically get locked in place, and the skeletal muscles will seize up. Now this won't last forever, of course, because eventually the proteins themselves, the actin and the myosin, they'll break down as part of the deterioration process soon after. It may take hours though, correct? Okay, a little grim there at the end, but thanks for watching that one. We made it all the way through excitation, contraction, coupling. Maybe soon we can get into naming some muscles. Check out some other videos if you found that one entertaining. See you for the next one. Bye-bye.